I'm Shriram Sundararaman, a fourth-year medical student at UVA, and this is the UVA MiniMed special podcast on COVID-19 Charlottesville. The MiniMed program dedicates one session every year to show its students about the groundbreaking research going on at UVA and how it may benefit its patients. Research is an important part of UVA's response to COVID, as it opens doors for the development of drugs that may shorten the clinical course of the disease or vaccines that may help stop spread the disease. I'm happy to be joined here today by Dr. Bill Petri, the Vice Chair of Research in the Department of Medicine at UVA, who actually started his career here as an MD PhD student. In addition to taking care of patients in the hospital, he runs a prolific research lab that studies the human immune response to several microorganisms, such as C. diff, and also spearheads international efforts abroad. Thank you for joining us today, Dr. Petri.
Oh, you're welcome.
Although multifaceted, one specific effort of his lab has been looking at the human immune response to the coronavirus. And we are happy to be joined here today by Alexandra Donlan, a graduate student in immunology in the Microbiology, Immunology, and Cancer Biology department here at UVA. Thank you for sharing your expertise with us today, Ali.
Happy to be here.
Great. So just to jump in, Dr. Petri, do you mind telling us in what capacity is UVA contributing to the research related to the human immune response to coronavirus, ranging from basic science research all the way to clinical trials?
Well, I think we're starting with clinical trials. Like the biggest advance is Patrick Jackson's work with remdesivir, which you're probably familiar with. And so he was part of the 1,000-patient clinical trial that demonstrated that remdesivir decreased days of hospitalization by about 5. So that's probably like one major thing. I think Amy Mathers, of course, has contributed with Mendy Poulter in the development of the PCR diagnostic test, which was so helpful as far as being able to manage patients in the hospital. And I think like from basic science standpoint, I think there's a lot of work going on vaccine development, and that includes Brent French and Craig Slingloff, who are using an adeno-associated virus vector vaccine development, and that includes Brent French and Craig Slingloff, who are using an adeno-associated virus vector, and Steve Zeichner, who is working on using a bacterial platform for a coronavirus vaccine, and then our work, which is using toll-like receptor agonists as an approach to vaccinate. And then the work that Ali can tell you about looking at immune dysregulation during COVID-19.
And do you have any insight on sort of how the closures and government shutdowns have impacted research in general at UVA?
It hasn't brought it completely to a halt, but it certainly has slowed things down, understandably. There's no scientific advance that's worth risking one's life for, and so I think it's made good sense to shut things down when we were at like the peak of the pandemic in Charlottesville. One of the advantages of like modern research is that most people have like very, very large data sets, and so there's always data to be analyzed, which is a nice thing. Probably wouldn't have been true like 20 years ago. And so I think for the most part, everybody is working through data that they already had, writing papers, planning grants, thinking about science.
And Ali, Dr. Petri did allude to your work a little bit. We've all heard in the news about the sort of scary cytokine storm that coronavirus can cause in patients. Do you mind explaining for our viewers, the science behind these cytokines and why are they specifically concerned in COVID-19?
Yeah, of course. So cytokine storm, or sometimes referred to as cytokine release syndrome, refers to this mass production of pro-inflammatory cytokines or proteins that help facilitate immune responses. And you may have heard in the news cytokines such as IL-6 or TNF-alpha. And so resulting from this, there can be serious complications and consequences, including things such as acute respiratory distress syndrome or ARDS, or multiple organ dysfunction or even failure. And so it has been observed that a lot of these cytokines have been seen elevated in severe COVID-19 patients all around the world, and causes of death here were consistent with cytokine storm as well. And so because of this, researchers like ourselves have a great interest in understanding how cytokine storm is contributing to the disease and how we might be able to target it to treat patients.
Okay. Do you mind telling us a little bit about the work that you're doing specifically in the lab?
Yeah, so I actually started out researching the immune response to C. difficile, which causes high levels of pro-inflammatory cytokine release similar to COVID-19. And during C. diff infection, it's understood that this pro-inflammatory response is pathogenic when in excess. So our lab has really been interested in looking into other immune mechanisms that can ameliorate this pathogenic inflammation. We've actually found that type 2 immunity, or immune responses that contribute to allergies and asthma, are actually protective. So my project has been studying this type 2 cytokine IL-13 and its role in promoting recovery from C. diff infection through immune cells called macrophages. And actually, it wasn't until recently that I'd started looking into the role of IL-6, another cytokine important for COVID-19, and its role during C. diff infection. And I found that when you blocked IL-6 activity in mice, they actually had worsened disease, which was really surprising to us. So since IL-6 is similar increased in severe COVID-19 patients, we have considerable interest in its roles during that infection as well.
Okay. And you said you have mouse models to study, these C. diff infections. Are there mouse models currently available to test the immune response to coronavirus?
There are some preliminary models. Our lab has interest in developing these models more further. So we currently have access to mice that are susceptible to SARS-CoV-2 infection, and we're working to amend this model so that we can look into different immune responses and how we can impact them to lessen disease.
Very cool. Thanks for sharing. And so, we talked about some of these promising research angles you brought up to suppress the immune response once patients already had the disease. But Dr. Petri, could you talk about any efforts at UVA going on to develop a COVID-19 vaccine or any sort of related efforts? I know you touched upon these TLR agonists and how it may be combined with a vaccine, but any efforts to prevent disease transmission?
Yeah, sure thing, Shriram. So there's probably the main thing is as Ali's alluding to, is to make a mouse model so that one can test for protection. And so Barb Mann and Brett Moreau are working with a lab, the Jackson lab has a mouse that overexpresses the human ACE2 receptor in epithelial cells, and that's the virus receptor, which is enough different in the mouse that mice are intrinsically resistant to the SARS coronavirus 2, but these mice should be susceptible. And so we're one of the test sites to see if this is in fact a good model. And we have the advantage of having a biosafety level 3 lab right in this building. And so one can do those experiments safely under negative pressure isolation. So that's the main thing is to have an animal model which one could then vaccinate and try to protect. Specifically the angle that we're taking on it is the way that vaccines work the best is if they have an adjuvant mixed with them, and adjuvants are things that can stimulate a better immune response. And so what we're using are adjuvants that are directed against toll-like receptors. And toll was originally discovered in Drosophila, making mutations in the fruit fly. They made a mutation that made the fruit fly susceptible to fungal infections. And they discovered they had knocked out a receptor called toll. And toll is one of the ways that your body can tell an invading microorganism from self. And so by mixing the vaccine with adjuvants that activate toll-like receptors, and it really activates the immune system because the immune system thinks that a bacteria or a parasite or a virus has invaded there. And so what we found with our work with an amoebic dysentery vaccine is we get a very robust response that Myresh Abhyankar in the lab has shown is able to induce both mucosal and systemic immunity. And that's probably important that you have IgA antibodies actually in your respiratory tract to protect you. And so we think that this combination of adjuvants should be able to do that for the SARS coronavirus, just as it does for amoebiasis.
I see. Okay. So sort of, as you've alluded to, the adjuvant sort of a alarm for the immune system to wake up and maybe spike their response.
Yeah, that's right. There's for example, like the new shingles vaccine, it's called Shingrix, is a recombinant protein, one protein from varicella zoster virus, and it's mixed with adjuvants that activate the inflammasome and also activate toll-like receptor number 4. And that vaccine is like 95% effective at preventing shingles, whereas the attenuated live virus vaccine is probably less than 50% effective. We usually sort of think of like an attenuated virus as being like the best possible vaccine, but it's an example of how adjuvants have changed the playing field, so to speak, for vaccines.
Mm-hmm. Yeah, and one question a lot of our viewers have is, when might a vaccine be available? And I know that's a hard question to ask, but just from work at UVA and your expertise, do you have any sort of insights on estimates for when a vaccine might be developed or what that process might entail?
Yeah, well, we're vaccinating mice now, so we'll have like some idea whether our approach is fruitful or not soon. I think that the vaccines are going to be most readily available are ones that are building on an existing platform. And so Oxford University and also Johnson & Johnson is working with an adenovirus-based vaccine that's already been shown to be safe in the case of Oxford in malaria vaccines, and J&J has been using it for a respiratory syncytial virus vaccine. So that'll shorten things, because that's one of the things that slows down vaccine development, is that it has to be very safe because you're giving a medicine to someone who's totally healthy. And so there's a very, very high threshold for safety.
Wow. Yeah, coronavirus has really put a halt to a lot of work around the globe, but it sounds like it's only created more opportunities for you to do more meaningful research. Whether it's from the lab bench or the patient bedside, we really appreciate all that you do to improving patient lives here at UVA. So thank you for your time. And to our listeners, thank you for joining us as well.